RNAs and virus genome sequence numbers. So in our lab specifically, we work with alpha viruses, and a virion is considered one virus particle, which is illustrated here. And so this is a kind of broad picture, I guess, of it, and here's a cross section. And within the virion, there's a single RNA strand, which is represented by this diagram here, figure 8. So the RNA is considered a positive sense RNA genome, and it has around 11,500 base pairs, or 11.5 kb. And it's important because it affects a diverse range of organisms, anywhere from vertebrates and invertebrates, such as mosquitoes, or even horses and humans as well. So it's mostly transmitted by arthropod vectors, and there's two groups of pathogenic alphaviruses to humans. So there's encephalitic, which is like neurological disease, and there's also arthralgic, which is like arthritis and things like that. And sadly, there's no available vaccines or therapeutics for these. So in our experiment specifically, we wanted to focus on Venezuelan equine encephalitis virus, or VEVE, and it's a re-emerging arbovirus within the Americas. And as you can see in this diagram here, it's located by the purple and distinctly marked, so that's where VEVE currently is. And then all these triangles and squares show a major outbreak or a major epidemic that has occurred. And it's mostly transmitted by mosquitoes, and like I said before, it's predominantly found in Central and South America. So the thing about VEVE is that it has similar symptoms to other arthropod-borne viruses. So that's also known as an arbovirus, and that can make VEVE very difficult to pinpoint and diagnose. And so the thing about this is that in humans, it causes flu-like illness, but more importantly, especially within children, it causes neurological disease for around 4 to 14% of people. And that's really important because a child that is born normal and have a happy life or something, if they contract VEVE and get neurological disease from that, then their life is going to be kind of harder to survive in society and function properly. So it's a really big deal that we need to look into this. And even though the mortality rate is pretty low, like I said before, the fact that it causes neurological disease is very important. And in equine, such as horses, it's, with some virus strains, it can be very severe, inducing mortality rates anywhere from 19 to 83%. So VEVE can be transmitted through two distinct cycles. The first being enzootic, which is always going on, and it's found within forests and it's directly transmitted between rodents, such as mouses, and mosquitoes. And the thing about this is that it can also, if there's a virus mutation that occurs, and there only needs to be one, then it introduces a whole new cycle known as the epizootic. And that is directly between horses and mosquitoes. And this one is really close to humans because humans work a lot with horses and whatnot. And it's also, the thing about epizootic is that it occurs mostly in epidemics. So you might be asking, well, how do humans come into play? Well, they can contract it from both the enzootic and the epizootic. They're considered dead endos, meaning that it can't be transmitted between them, but it can be contracted from mosquitoes and different transmissions. So let's talk about TC83, which is a live attenuated vaccine. Live attenuated basically means that it's just a weakened version of the virus. The thing about this vaccine is that it's not approved for human use, but it's used to vaccinate horses. And it also allows us to work with VEVE at the BSL-2 level, or biosafety level 2. So now some of you may be asking, well, how does it travel through the mosquito? Well, first, the mosquito has to bite a rodent, such as a mouse or something, or a horse. And the mosquito ingests the VEVE-infected blood through the proboscis, which is here, and it infects the posterior midgut, which is located here. So then after that, it disseminates into the hemocell, which is the mosquito circulatory system, and it circulates within. Then the virus then disseminates into other body parts, like the head or the saliva glands, which are more important because they produce saliva. And then from there, depending on the mosquito, this can take anywhere from 2 to 14 days. So now you may be wondering, well, how does the mosquito combat this attack? RNAi, which is the most important antiviral pathway in mosquitoes, is of debatable importance within vertebrate antiviral defense, but it's important in mosquitoes. It's activated by double-stranded RNA, which is here. And it's produced when the virus is replicating its RNA genome. And there's three pathways that lie within mosquitoes. There's siRNA, which is the most mutation of RNA, or the virus mutation, sorry. So as you can see within the first replications, we already see hints of mutations. Within three, there's two mutations. And as you can see, as it keeps on replicating and replicating and replicating, by the end, you're left with a very, very diverse population. And that's important because we need to understand how all those mutations interact together. Do they benefit the host? Do they cause harm to the host? So that's why it's very important to investigate. So the sequence specificity of RNAi may influence virus genetics. That's what we're kind of believing. So that we found this through a study in West Nile virus where they concluded that genome regions more intensely targeted by RNAi had greater genetic diversity. So our hypothesis was that there will be increased virus genome sequence variation within mosquito cells that have a functional RNA response. And that's important because it can affect host tropism, disease severity, and resistance to therapies. And most mutations are expected to be deleterious or neutral. So I'm going to kind of go through here in figure 8 and kind of show like how it might mutate within itself and kind of how it affects the host and post-replicating or producing offspring or so. And in this example, we'll be using like humans. So in the first mutation rates, the black represents a lethal. So say a baby being born without a brain or something like that. They probably won't survive and reproduce or so. So they'll just kind of die off, which we can see in the last figure here. Then you have neutral mutations, which is just kind of like, you know, maybe you have like an extra finger or something like that. It's kind of just neutral. It doesn't harm you and it doesn't add to your equipment as well. So as you can see, those they'll just kind of reproduce within their own society and just kind of stay the same throughout. And then you have deleterious mutations, such as being born without an arm, being born without legs, something of that nature. And as time goes on, those people tend to become less and less and reproduce less often. Whereas a beneficial mutation, such as genetics, maybe you're a great basketball player or something like that, like all these freak genetics or something, those people produce more and more and you get very high amounts of highly beneficial people. So in our specific experiment, we focused on U4.4 or C710 mosquito cells, and they were infected with TC83. And the reason why we chose U4.4 and C710 is because U4.4 has a functional RNA response, whereas C710 don't. And the purpose of that is to see if there is an increased genetic variation in the virus isolated from the U4.4 cells. And we expected that there would be reduced diversity within the C710 cells. So I'm going to go through our methods that we use to conduct our experiment. So the first thing that we needed to do, first order of business, was to isolate RNA by itself. And we get that from the virion. Since the RNA is entrapped within the virion, we needed to break open the envelope just to get the RNAs by itself. So from there, these small RNAs derived from the cell were then sent directly for sequencing. But the virus RNA still needed to be amplified before we could sequence it. So then we performed reverse transcription, where we inserted a primer, such as a random hexamer, and it bonded to a specific part of the RNA. And after that, it copied what it saw in the RNA strand, and it produced cDNA. Then from cDNA, it performed PCR, and it basically took that copy and made 2 copies, and then from there, it amplified and amplified and amplified exponentially. And then after that, we needed to confirm that the PCR worked, and in order to do that, we needed to perform gel electrophoresis. So over here is one that we conducted within our own experiment, and as you can see here, here's a ladder indicating how many base pairs there should be, and over here is our virus genome sequence. And the thing is that we needed to determine that each part of the strand had a specific number of base pairs that we were looking for. And in order to guarantee that throughout the entire strand, it was kept through, maintained the amount of base pairs needed, we divided it up into 9 different segments. And that can be shown here. So this would represent probably the first section, the second section, and so on and so forth. So the first one, as you can see, within our own example, the first one had the correct amount of base pairs, whereas the second one didn't, the third and fourth kind of did, the fifth kind of did, but the sixth didn't, and the seventh and eighth did. So the thing about the ones that we're missing is that we probably had to go back and PCR amplify those as well because of the fact that they didn't show up until increases. So then we performed PCR purification and then the isolated DNA for simple sequencing. So in the future, we want to work with blockhouse virus B2, and it's a virus protein that blocks all 3 RNA pathways and inhibits dicer cleavage of double-stranded RNA. And we'll be inserting this into the TCA3 genome to see how this affects the populations of mosquitoes, mutations in mosquitoes, and mosquito cells. I myself didn't handle any of the animals and received all information presented to me by my mentor. And I'd like to thank Tiffany, Dr. Forrester, the UTB Department of Biology, and the Department of Microbiology and Immunology. There's Twig, Scott, Mosquito Center for Environmental Health and Medicine, Dr. Young, Robert Chairwoman Trust, and the Center in Environmental Toxicology. So we're excited. Are there any questions? So you're asking basically how they can determine and differentiate between VEEV and flu, correct? Okay, so the thing about VEEV is that it's, like I said, it's very similar to the other arthropod viruses, which makes it kind of difficult to necessarily pinpoint. But within, I guess, the VEEV specific strand, they can I guess, take a sample or something and kind of send that for sequencing, I guess, to determine the actual sequence. Does that answer your question?